Welcome. We'll be starting shortly to allow a few more attendees to join. Hi, everyone. My name is Angie Hofnagel, and I am the Marketing Director at Austin Pharmaceutics. I'm excited to welcome you to the second session of Austin PX Live. The series gives you an inside look at who we are, what we do, and how we partner with innovators across the industry. In today's session, our Chief Scientific Officer, Dr. Dave Miller, will share how Austin Pharmaceutics builds a strong foundation in early development through molecule characterization, analytical expertise, and preclinical formulation strategies. We'll begin with a 20-minute presentation followed by a live Q&A. Please submit your questions at any point throughout the session. Now, I am pleased to introduce Dr. Dave Miller, scientific leader, Kinetisol co-founder, and an expert in formulation strategy. Dave, the floor is yours.
Thanks, Angie, and thank you all for joining us to episode 2 of our Austin Pharmaceutics Live event series. If you joined us for the first event, it was a lot of fun. We covered who we are as a company. Austin Pharmaceutics has got a unique history for a CDMO, starting out as an innovator of our Kinetisol platform, focusing on bioavailability enhancement and partnering primarily with large pharmas, to migrating to becoming a sponsor ourselves and developing our own clinical pipeline of products, and then being very successful in that mode, and then making a successful exit, partnering those assets out, and then repurposing ourselves as a general CDMO. So we have a very unique history as a company, and in the first session of this live event series, I covered our history and how that shapes us as a company in terms of our scientific depth, our expertise, how we operate as a CDMO, the commitments to our clients and our culture. So if you didn't catch the first episode in this series, I really encourage you to find the recording on our website and watch. It really sets the stage for all the subsequent episodes. So today for episode 2, the title, as you can see, is Navigating Complexity. Focusing on early days of a new molecule and our approach for analytical and preclinical development for new compounds. But before we get into the details of that, I want to just quickly reiterate that history that I've just described with this slide, looking at who we are as Austin Pharmaceutics, just at a glance. I like this tagline here, where complex challenges meet tailored solutions. That really describes our mentality. Coming from a former sponsor ourselves, we look at every new molecule as a valuable asset, one that needs to be handled with care, understood, developed, not just a boxed solution for a molecule, but actually coming up with a tailored solution for that molecule that meets not only the challenges of the compound, but also our clients' specific needs, the program's specific needs, just tailoring our approach for all the various facets involved in developing a new molecule. And it really starts with our scientific depth. We were a company that was founded on a commitment to excellent science and engineering, and letting that drive drug product development towards an unmet therapeutic need. And we've been doing that for over a decade. So we have a long history of proven success and expertise in these areas. So as I mentioned at the front, we started out focusing on amorphous solid dispersions. That was our entry into the market. And that's what emerged into bioavailability as a general problem, of how we solve bioavailability problems, how we characterize bioavailability problems up front, and how we characterize those solutions, and how we extrapolate from those early characterizations for long-term success. And then along the way, I think anyone working in this industry has to become, in today's climate, a potent compound handling expert. It's not just enough to have delivery challenges, it's also handling challenges, health and safety challenges that we've built up our capabilities in terms of containment and procedural handling to handle any molecule that comes in the door, not just development and delivery challenges, but also potent handling and manufacturing challenges. We know how to handle all of those issues. As I said, we started with our kinetosols, our proprietary ASD platform, and you know, springboarding off of that to developing elaborate and systems for handling any and all problems, not just bioavailability problems, but other issues involved in the development of oral dosage forms. So we've had 15 plus years of experience in dosage form development and GMP manufacturing, and we've worked on over 100 molecules to date. And that number is rapidly increasing since we rebranded to a general CDMO in the last few years. So I've already mentioned our sponsor history. At peak, we had 6 clinical assets in the pipeline, and that we were developing ourselves not just through the CMC, but also doing preclinical, clinical, regulatory interface, clinical operations. We've been there, we've done it, we've been operating on investor funding. We know how important each milestone is. We understand burn rates. So it's very unique to have a CDMO who's been in that sponsor seat and can really appreciate all of the challenges that our clients are facing, not just technical challenges, but business challenges as well. And we can not only provide advice, but it's empathy, it's understanding what our clients are dealing with and being a true partner in development, solving their challenges and meeting their deliverables on time with open communication and transparency. And so, as a sponsor, we worked with a number of different CDMOs, um, great partners, not so great partners. We know what the habits of a good organization, a good CDMO are, and we know what the habits of a poor CDMO are. So when we rebranded as Austin Pharmaceutics and we became externally facing, our we set up our systems and our culture to nurture the good habits of a good CDMO partner and eradicate the bad habits that we saw in our bad CDMO partners. So again, starting with scientific and engineering expertise, but building a culture and a framework and systems of a great CDMO that go beyond just the technical, but into project management, into culture, into the interface with our clients and other parties involved. So we set up systems that are designed to be fast and flexible. We understand how in early development things can change quickly. Drug substance properties can change, delivery times can change, preclinical needs, study dates, you name it. We've seen it all go wrong. You think you can, as soon as you're predicting what's ahead, and you think you've got a good sense for what's coming, the landscape can shift quickly. So we understood the need to develop our systems to be fast, flexible, transparent, and also you know, as I said up front, what's key is that we look at every molecule as a new challenge for designing a custom solution. So we let the data drive, and that's a common theme throughout this presentation. Collecting as much data, molecular characterization, and letting that data, actually just following the data along the path of the most efficient means for getting the molecule through preclinical development into the clinic. And by letting the data drive, it's a platform neutral solution. We're advising our client as to the best approach for their molecule and not boxing them into some proprietary platform. So again, this slide just, again, reiterating, we're oral dosage form development experts. We were founded on solving the most challenging bioavailability problems that the industry was facing. And from that beginning, we've really layered our capabilities to cover all manner of different problems. We've, at this point, 10 years in, internally and externally focused, we've seen just about everything that can happen in oral dosage form development program, and we know how to solve these problems. We have a long track record of solving the problems. But if with every new molecule, it starts with preclinical development. And preclinical development starts with complete molecular characterization, building a robust pre-formulation package, letting that information drive technology screenings towards developing a delivery system for that molecule to satisfy early PK, GLP tox needs, enabling an IND. And if that moves into formulations enablement, we've got all of the tools and the expertise to satisfy in the early preclinical stages. And then moving forward, when we're going into manufacturing and developing an oral drug, we've done everything from simple solution suspensions, directly compressible tablets to modified release tablets, capsules, whether that be blended or granulated formulations, or just API in a capsule, trying to get into the clinic quickly to get a safety efficacy read on the molecule. We've supported it all. Modifying release to achieve a specific PK target. We've done that, so we can support drug product release from anything from immediate to modified, delayed, pulsatile. We've designed dosage form to achieve a number of pharmacokinetic endpoints that require fairly sophisticated formulation development. And then in simple cases where all you need is an immediate release tablet, something that adequate tensile strength and rapidly disintegrating. We've done many programs like that. And our services span from not just preclinical, which I'll describe today, but all the way through phase 2 manufacturing support. And so we've got it in terms of dosage form design, there's a depth of expertise, but like I said, as a sponsor, we understand burn rates. We understand milestone-driven funding events. So in a lot of instances, it's about getting into the clinic very quickly or supporting rapid manufacturing for different modalities. So we design our programs and our dosage forms to be phase appropriate, to get into the clinic quickly, get initial safety reads, efficacy reads, in support of, first in human trials, orphan indications, breakthrough designations, whatever the program entails. We've got a system set up to provide deliverables along those timelines. Okay, so let's start with our approach for early preclinical development. So as I said, it starts with a foundation of molecular characterization. We want to know everything about the molecule that we can. We want to identify any red flags and let that data really drive the formulation development process, entering into GLP tox studies and ultimately delivering the data set that's IND enabling and moving the molecule into clinic. So our approach with a new molecule is to do comprehensive characterization and pre-formulation testing. The bullets on the right really describe a majority of what we would do. In that instance, these all should look very familiar. But starting with in silico analysis, it's very easy with the right software to plug in the molecular structure and start to get a sense for what the key properties are roughly, and begin identifying any particular red flags or areas where we need to dig a little bit deeper in terms of our pre-formulation characterization. It's simple, it's easy to do on the front end. So we start in silico. And then from there, doing all the characterization that affects solubility, permeability, things like pKa, log P, looking at intrinsic dissolution rate of the bulk drug substance, understanding crystallinity in terms of polymorphism, thermal analysis, melting point versus glass transition temperature, looking at the moisture content of the API in terms of hydrate, residual solvents. Those can be very impactful for downstream processing. Hygroscopicity of the drug substance is obviously key, doing dynamic vapor sorption analysis, sorption, desorption events, understanding how hygroscopicity could also impact stability, manufacturability down the line. Characterizing particle size for every lot of API that comes in, understanding how particle size distribution is changing, how that may affect performance and manufacturability. Likewise, crystal habit. If we're dealing with needles, cubes, and how the habits may change depending on the crystallization process, like in early preclinical development. A lot of the final crystallization steps are not always set in stone. Those can change, and with that, so too can the crystal habit and the particle size distribution. So very important to understand that on the front end, as that has tremendous impacts downstream. Obviously, solubility profiling is really key. Not just looking at sort of simple aqueous media, buffers, but also biorelevant media. The pH solubility profile, organic solvent solubility, if we need to go an enabled route and we're evaluating spray drying or other solvent effects, we need to understand what solvent systems are going to be most effective. A lot of the compounds that we see have limited organic solvent solubility as well. So if we need to go down that enabling path, we need to understand how organic solvent challenges, the solubility challenges of an organic solvents might lead us down the path of a solvent-free process. Looking at also the amorphous solubility, liquid-liquid phase separation. I'll talk more about that later, but it's really important to understand how to do the amorphous solubility, see how an amorphous approach how that solubility enhancement relative to the crystalline form can enable or the bioavailability enhanced formulations, or if a crystalline solubility, if the delta is not that large, you may be able to proceed with a crystalline form in a specific vehicle or with particle size reduction. Biorelevant partitioning studies are very important as well, not just about getting a general log P, but also looking at biorelevant media and how how the biorelevant media may affect partitioning. Important to understand early. Obviously, stability is the cornerstone. If you don't have a stable formulation, stable molecule, you don't have a developable asset. So we want to fully understand the modes of degradation of the API and mitigate or identify any potential places for compound degradation as that affects formulation, and mitigate those early. Design the formulation system to overcome chemical instability issues. Also looking at permeability, not just Keco-2 assays, but also transmembrane flux. As I'll mention later, we have pion flux systems that we bring to bear on most projects. There's also other related assays like Using chamber that we've been a part of to help characterize the permeability of a molecule. So finally, you take all of this in silico and experimental data, and you culminate that into a developability assessment that, again, data-driven. This is what we understand about your molecule and its potential challenges, and here are the formulation pathways we recommend through preclinical studies and extrapolating beyond. Okay, so we've laid the foundation. We're building a custom home for your molecule that'll house it through development. So it starts with a preclinical foundation, but next we need to build a framework where we can evaluate these formulations. And I like to think of the biopharmaceutical tools as that framework. With that established, we can design formulations within that framework and understand if they're going to meet the needs of the program. So this is a combination of in vitro tools, in silico modeling, and our preclinical development expertise. So in terms of in vitro, I've already mentioned assessing amorphous solubility through solvent shift and LLPS assays. Biorelevant dissolution is obviously an important tool. USP2 basic dissolution apparatus is still important. We primarily focus on our pion systems, using our micro and macro dis systems quite often, but also looking at the sort of traditional pH shift methods, shaker flask methods, disso and syringe, kinetic solubility studies. Depending on what the program needs, we have all of these various tools, or we can characterize the in vitro release of the drug substance itself and in formulation. Then once again, solubility, permeability go hand in hand in predicting bioavailability. So we're looking at transmembrane flux and other permeability assays. And then finally, as an extension of amorphous solubility, we can look at concentration enhancement through excipient mediated solvent shift methods. How does an excipient influence the amorphous solubility, and utilize that information in the design of the formulation. And then in silico modeling, we doing in silico that's informed by a lot of the preclinical information and the in vitro information. We've done this using proprietary software like GastroPlus or SimSip, but also open source models as well, like PKSim. And then, obviously we know the predictive power of these models, but also integrating into the models the data that we're generating to make them more robust. And we keep sort of iterating through these models as we collect more data so that refine them and help better direct development. And finally, this is sort of capped off by our own preclinical formulation expertise. We've been involved in countless preclinical development programs with large and small pharmas. So we understand how to take the data that we're generating through preclinical development and through our biopharmaceutical characterization and assimilate that information and use it to decide what is our formulation pathway, whether it be a simple aqueous or non-aqueous solution or suspension formulation, or if we need to do more advanced formulation development for bioavailability enhancement to increase exposures to establish the NOEL and the safety limits as you move towards IND enablement. So we've got our foundation and our framework. Now let's put up the walls, the fixtures, the plumbing. Let's design this delivery system for the molecule through formulations development. So the process, just again, once we know our molecule, we've got all the key data, we've determined all the red flags, we've let data drive our formulation screening approach through simple or more advanced systems, and we know where we're sitting in terms of developing a basic tox formulation or something more advanced. We usually start out doing some early PK screening to verify the formulation approach, and then we will, further optimize that delivery system, looking ahead towards tox studies. Once again, exposure is key. We have to establish that no EL, so we have to drive exposures to, a 50x safety margin, so that we can move forward through clinical development. So optimization is typically about maximizing exposure. So not only how we design the formulation of the API, but also the vehicle for administration, and doing this without consuming a lot of API and in rapid turnaround to meet study start requirements. And then once we've developed the formulation, there's a tech transfer, if you will, between the development of the dosing system and the site that'll actually conduct the dosing. So we've found very close interaction between ourselves and the study site is critical for success. It's not enough to develop the formulation and the dosing protocol, hand it over to the client, walk away. You really need to closely interface with the study site to ensure that the dose preparation and the dosing procedure are done properly to ensure the integrity of the study. So when we go through this whole process, we've invested in understanding the molecule, creating a framework of tools to help predict performance, defining the right formulation approach, developing a robust formulation and a dosing procedure. You've made a significant investment in early phase success. You can be confident that what is being provided into preclinical studies is effective, and the results of those studies will support your entry into clinic in terms of establishing the safety of the molecule. And if we run into those issues with achieving elevated concentrations to establish safety margin, and we have to go to enabled formulation screening, this slide just illustrates that sort of hierarchy of that process. So once again, starting out with understanding the molecule from in silico pre-formulation studies, having the right methods in place to evaluate these formulations, and then from there, taking sort of a shotgun approach and different means of enabling bioavailability of the molecule, starting, particle size reduction, whether it's micronization or if we need to go the nanoparticle route, doing lipid formulation screening or non-aqueous based vehicles to deliver the drug in a solution form for supersaturation, or if we need to go to the ASD route, doing some pre-formulation work to understand what are the most effective excipients in terms of maintaining the amorphous form in a stable amorphous physical form in suspension, and also promoting supersaturation and driving exposure. So we do some pre-formulation front-end work to see what are the most effective polymers for satisfying suspension stability and exposure. And then from there, we can branch out into our technologies. So like most CDMOs, we have spray drying melt extrusion, but we also have our Kinetisol platform, which is unique to us. And Kinetisol, as we found with our work with Boehringer Ingelheim and Lilly, we've got some really great public examples of how we enabled their molecules to achieve exposures that weren't achievable through spray drying. So between these three approaches, Kinetisol, spray drying, melt extrusion, we can develop an optimum ASD for highly insoluble molecules that where an ASD is required for successful tox studies. And then finally, cyclodextrins, another effective tool in a preclinical setting for increasing solubility and exposure. And we can screen cyclodextrins by traditional routes, but also Kinetisol as a means of creating solid-state inclusion complexes is very powerful. So the frictional processing of Kinetisol is suitable to create solid-state inclusion complexes in a very efficient non-solvent approach, and that can be useful as well for creating increased concentration of drug in vehicle to promote tox dosing and the achievement of high exposures in tox species. Okay, so we've set the framework of biopharmaceutical testing. We've filled in the walls, the fixtures, the house is built, roof's on. Now it's the inspection. How is the formulation approach working? And that's the the non-GLP PK studies. It really comes down to that milestone. You're dosing these formulation concepts, seeing which ones are most effective in terms of achieving target exposures. And we as a company have conducted dozens of these studies for ourselves and for external partners. We have over a decade of experience of executing these studies. We've seen every way that they can go wrong. We know how to identify those issues early on and how to avoid them. So if we provide this as a service to our client where we will interface with the study site. We'll do that transfer of formulation knowledge, dose preparation, and ensure that it's done well at the study site. So specifically, we're talking about oral PK studies in canines and rodents. And this is typically done by gavage, but also solid oral dosing in dogs or miniaturized capsules in rodents. We've also been a part of developing IV controls as needed and where possible. We do comprehensive protocol design and study planning, working with the study site. We don't just leave it to the study director to propose the study. We work hand in hand with the study director to design the appropriate study. I've already mentioned optimization of the formulation and the dosing parameters is critical to study success and integrity. And that's where we focus a lot of effort and where our expertise really adds to the value of a study, but also mitigating any issues or wasted time or resources. And then obviously these studies involve in-life observations, the bioanalytical and pharmacokinetic analysis. So, and the benefits to our client is when we're interfacing with the CRO, you've got streamlined communication with the study site. It's one point of contact to manage the entire study and ensure the outcome. And like I said, we've seen the ways that these studies can fail, so we can mitigate those failures. And it starts with protocol and dose preparation, but it also goes through in-life troubleshooting. It's not uncommon for study directors to contact us with some observation that may have an impact on the study. And then based on our experience and expertise, we can guide them based on that observation to a revision of the plan to continue the study and ensure its integrity. As far as deeper insights go, it's easy to just look at the means of the PK, look at the PK results, the means, the statistical analysis, but you find you get much deeper insights when you look at the individual's plots, when you look at the in-life observations and match those up to the pharmacokinetic outcomes, and really start to understand, the performance of the dosage forms holistically. Did it cause emesis? Was there loose stool? At what time points? How did those observations seem to affect the plasma levels? So this comes from years of doing these types of studies and understanding the importance of not just looking at means, but looking into the finer details of the study and to have a better understanding of its outcome. And then finally, when the results come through, we reiterate back to our PBPK models to expand those further and to make them more robust, and it will ultimately help to refine our development strategy long term in having these robust models. So finally, just in closing, why partner with us? Well, like I said, we were a company that was founded on scientific and engineering excellence, specifically for bioavailability enhancement. And as we evolved as a company, that same excellence has expanded to all things CDMO, not just on the technical end, but also project management, regulatory, QC, QA. We're really committed to excellence, and we're committed to customer satisfaction. So that alone is a great reason to partner. But beyond that, our approach is focused on understanding the molecule, full characterization, designing a program meant to provide a tailored solution that's fit for purpose for that molecule, for that client's immediate needs. Not to box them into some standard approach, but to really listen to the needs of the molecule and the client and find a pathway forward that's fast and efficient, gets them into the next milestone as quickly as possible. So it's high quality science with speed and efficiency, transparent communication. We have the industry's best project managers, well organized, highly transparent, allow free access to the technical team that's actually executing the science, so that they're working hand in hand with our sponsors to deliver on their needs. And then finally, proven outcomes. We've been at this specific uh, place for the specific activity of developing complex dosage forms and less complex dosage forms, but quickly and efficiently for over a decade. And we have a long history of success with clients of all size and at different phases of development. So our culture, our history, our scientific commitment, they all lead to a long list of success that has frankly results in loyal clients that come back to us no matter what the challenge. That for the appreciation of the science and the service. And with that, went a bit long, but I hope we have plenty some time for questions, and happy to answer any questions y'all might have.
Thanks, Dave. Yeah, we are running a little bit behind, so I'm going to just pick two questions for you. Um, and then to the audience, if you have questions that were not addressed, please feel free to reach out to Dave or myself. So the first question is, why is formulation strategy so important in GLP toxicology studies?
Yeah, I think it's a good question. A lot of clients and not just clients of ours, but other folks that I've communicated with in the industry, and then they don't pay close enough attention to the tox formulations, kind of like micronize the drug, throw it in some standard vehicle and dose, and or maybe go a little bit beyond that and develop a tox formulation that should be good enough. And then you get into a tox environment, you start escalating the dose. You're not quite achieving the exposures you need to establish safety margins. As you escalate, you're seeing a plateau, and now you realize that the tox formulation that you rushed into the GLP studies is inadequate. And now you have to stop the study and go back and do formulations development to increase exposure. So we've seen that a number of times where as the dose is escalating in tox and the plasma concentrations aren't increasing, you've got to stop and reformulate. So you're experiencing delays, lost time, lost money. It's a better approach to, on the front end, in that biopharmaceutical framework that I've described, really evaluate the formulation options to ensure that what you take into a tox study provides adequate exposure over a broad dose range so that you can ensure you can achieve whatever systemic concentration you need to achieve to establish the safety margins. So we've had to come in as the tox rescue team a number of times and develop bioavailability enhanced formulations that not only drive exposure, but are stable in the dosing vehicle. And I'm talking physical, chemical stability that can be accurately dosed. I think I failed to cover, but when we develop a tox formulation, whether that be a powder for constitution or a solution or suspension formulation, the characterization doesn't end at biopharmaceutical characterization. It's also the analytical characterization. Is the drug chemically stable? Is it physically stable? Can dosing by gavage be done reproducibly and accurately? So we've come in on a number of programs after the fact, done this sort of systematic preclinical development, and got the GLP tox program back on track, but not before the clients had suffered months of delay and lost time, money. And like I said, if you're a small biotech running on investor funding, getting to those milestones quickly and efficiency is absolutely critical to the health of the company. So that's why investing in preclinical development and tox formulation on the front end is so important. Don't overlook the importance of your tox formulation, or else you could really suffer delays and financial setbacks that can be really difficult to recover from.
Thanks, Dave. And last question, which molecule properties most often cause setbacks in early development?
Yeah, from my experience, chemical instability and solubility. Those are the two I've seen most often, and that's why we like to very early on assess chemical stability comprehensively, looking at not just solid state, but also stressing the molecule through different modes of degradation, excipient interactions to really verify that ultimately the formulation, the form of the drug and the formulation are chemically stable. A chemically unstable compound is not a developable asset. That typically only that's very early, usually, maybe pre-candidate nomination, but it's important to fully understand the chemical stability of the molecule in order to green light further development. And then from there, uh, poor solubility tends to be another, as I already explained in detail. You've got a compound that's solubility is lacking. You feel like you can overcome it through some basic formulation approaches, but when you get into the actual dosing environment, you find as you escalate the dose, that solubility problem becomes magnified. So you really have to bring enabling technologies to bear to get the molecule over the hump and into a mode that can be delivered to achieve the target safety margins. Yeah, that.
Okay. Well, thank you for those insights. And thank you to all of the attendees for joining today. This concludes the second session of Austin PX Live. Feel free to scan the QR code to register for our next session. The live series number 3 will take place on September 25th at 11 a.m. Central Time, and we would love to see you all back for that session. Thank you again for joining us, and have a great rest of your day.
Thank you all.